Assalamu alaikum. Good evening, everyone. This is Kashfia Shams. Welcome you all in our today's webinar on drug delivery systems, arranged by ACS Student Chapter, University of Dhaka. আমাদের চ্যাপ্টার সবসময় চেষ্টা করে আন্ডারগ্রাড স্টুডেন্টদের জন্য রিসার্চ কেন্দ্রিক এমন কিছু করার যা তাদের অপশনস গুলো তাদেরকে এক্সপ্লোর করতে সাহায্য করবে, তাদের প্যাশন গুলোকে খুঁজে বের করতে তাদেরকে সাহায্য করবে এবং হোয়াট অ্যাকচুয়ালি ইন্টারেস্ট এই বিষয়গুলো তাদেরকে খুঁজে বের করার জন্য সবসময় তাদেরকে সাহায্য করবে এমন কিছু করার। তো এরই ধারাবাহিকতায় আমাদের আজকের আয়োজন ওয়েবিনার অন ড্রাগ ডেলিভারি সিস্টেম। বর্তমান বিশ্বে যার সিগনিফিকেন্স আমরা আমাদের আজকের স্পিকার কাছ থেকেই শুনবো। এবং today we have Dr. Mohammad Nurunnabi as our speaker, who is currently an assistant professor at the University of Texas at El Paso. We will be taking questions at the very end of our session. So if you want to ask any question, please stick to the end, or you can leave your questions in the chat box and I will take your question to the speaker at the very end. So sir, I won't be wasting any more time and the floor is all yours, please.
Thank you, Kashfi. And, it was surprising to get an invitation from the EU. And I had no idea who are the people are, contacting me, because I have very few contacts at DU, unfortunately, because I was not a student of DU and, so not been in contact with people over there, not networking or connecting with them. So it has been always privileged to be there and, getting an opportunity to be able to speak about my research. And since this, webinar is very much focused for the undergrad student, I will try to keep it low key. Probably there's some terminology or something that might be hard for you to understand. So we will have maybe 15, 20, even longer, as long as you need to, or as many questions you have, we can answer at the end of the seminar. Basically, I'll be talking about, what are the chemistry behind of drug delivery and what is drug delivery? Like, is it delivering a drug from, Chittagong to Dhaka, like Yaba, or is it a delivering drug from, the Bangladesh border to the mainland? Or is it a drug delivery that drug is helpful for the body from side of administration to side of action? So, regardless of where you deliver the drug, for example, delivering through orally or injection in the muscle or injection in the vein, it has to go to the site of action. So when you say we have a disease, right? So for example, lung cancer. So that means the cancer is located exactly in the lung, not in the heart, not in the kidney, in the lung. And that doesn't indicate that whole lung is a cancer. Maybe, a very tiny part of the whole lung are cancer cells. So it is very important for us to be able to deliver the drug exactly on that particular cell of the organs, not other part of the organ, not other part of the body. So to be able to do that, it needs lots of effort, expertise from different backgrounds of people like chemistry, pharmacists, biologists, and definitely clinical scientists. So I will be, talking about maybe one or two projects to give an idea how we do that and what is the role we play in a drug delivery system or in translational research, where you develop something in the lab and then push forward to bring that to in the industry and finally to the clinics or the patient bed, right? So let us look at the value chain, which is the stage of the drug development or phases of the drug development. So target identification is the very early stage of drug development. So where a chemist play with maybe thousands of molecules is called shortlisting, has more targeting efficiency, specificity to that specific disease or specific, target site of the disease, right? So then you have to be able to find the lead, because you can't, work with 10 molecule, and finally bring one drug, because, every molecule, like it takes maybe 5 to 10 years to be able to, uh, develop and do the preclinical and clinical study, and finally do the NDA filing in FDA. So the smaller number of drug you kind of identify as a lead, then it's gonna save your time, it's gonna save your lots of money, right? So in lead identification, the biochemists are, responsible for narrowing downing, the number of molecules that, there is a risk for identifying one. And then candidate selection, right? That means, which candidate you want to, move forward with the formulation, because now you are moving to preclinical development where you're going to work with animals. In candidate selection, mostly we do, the cell experiment, or in vitro experiment, and preclinical is all about in vivo, which is small animal like mouse, rat, guinea pig, sometimes monkey, or even the pig. And then clinical development, which is, the phase 1, 2, 3, 4, like clinical trial, where you do the toxicity study, you do the biosafety, you do the therapeutic study, dose optimization, like how much you need at a time, and how many doses you need every day. And seeing like if the drug has the toxicity in a long term and short term administration, maybe you can working on that space. So, in our lab, or in my expertise, what I do in the process of drug development, or you know, you know, translating a drug from bench to bed, which is lab to clinic, we basically work on candidate selection, and then preclinical development, and sometimes we also move to the clinical development if in case we find something really exciting that, and we also have the capability of doing that. For example, millions of dollars of funding, industrial collaboration, and collaborating with hospitals. So those are the process we get involved. And NDA filing, that you need, definitely an active collaboration from a pharma, or funding support from the government. For example, the Moderna vaccine is being developed by the US government funding, right? Whereas Eli Lilly or Pfizer, they did not get government funding support because they're a large company. They have more money than probably the government. Anyway, that's the different story. So what I was trying to say is, in our lab, or in my expertise, we are not part of drug discovery, or the late stage of drug discovery. But we are mostly part of significant stages of pharmaceutical development. So then, I will be talking about on these three stages, with examples of the projects that we are doing, or we have been doing in our lab. So before going to the real work, I want to give you a real scenario of what happens, or what is the gap between the academic research and translational science. So if this is a paper published in 2003, 17 years before, but it has been summarized of, almost 2,500 articles, and those are the articles are published in a highly reputed journal. For example, Science, Cell, Nature, and Journal of Biological Chemistry, or Journal of Clinical Investigations. So I'm not talking about other there are millions of other journal which are maybe 10, 5, 20 impact factor, but those are not as kind of prestigious as those are. Anyway, so the authors identified 2,500 research article published in 1970, uh, 1979 to 1983. Those 10, how many years? Like 4 or 5 years, right? 4 years. And then those are they selected only 2%, which is 500 articles, which is they claim, the author claims, they have high potential of translation, right? And then um, finally they narrowed down to 100 article that shows resulted in clinical trial. That mean those almost in the late stage of the drug development, right? So they did already human trial. And finally, um, uh, you know so finally, after 10 years, they identified that only one of 2,500 articles, only one of the drug came out uh, as a drug. So can you imagine, there has been 2,500 project, and only one projects developed materialized as a product, as a drug, which is angiotensin 2 converting enzyme inhibitor, AT2. So I'll be talking about a lot about angiotensin 2 today. So that took 30 years. Okay, that took 30 years after publishing the paper. So think about it. So there has been 2,500 projects, and only one project's materialized as a product, and it took 30 years since the author published. So how many people, how many millions of grant, and how many years it took effort to develop only one product. So this is a real scenario where when we do something in academia and publish a paper, it's really, really premature, or I would say immaturity, to claim that we develop a drug, we develop a product. And it doesn't happen in drug delivery system or drug development. Whenever you are talking something about human health, that is totally different and very complicated. But when you're talking about, okay, we develop 5G or 6G today, you can bring the product in market next day. If you claim that I brought develop iPhone 13, or something better than iPhone 13, people will jump into it to buy it because that has no direct effect on health. But when you're talking about something that related to health, it could be drug, it could be any vaccine, or therapeutics, or whatever that is going into your body, that is serious concern about the human health, and you cannot risk human health without doing for a lot of studies in different states. So that's why it's very complicated. For a scientist like us, it's really would be lucky for seeing that at least one of my product is using in clinic, or the patient is getting benefited. It doesn't happen most of the cases. Think about it. There has been 500 articles, only one got published after 30 years. So that means only maybe 1% or less than 1% authors were that lucky to see their product in the market. So I'm saying this story to just give an idea of the young folks that when you're doing science or doing research, take it as a learning, take it as a the generating knowledge. Don't take it as a challenge of developing a drug, or claim that in social media, oh, we published a paper where we develop something, or we develop a drug, which is not true, because this is maybe other 99.996% which has been failed, and that most likely gonna be. All right. So, uh, this is a real scenario of what happened when you deliver a drug. We published this paper in 2019, last year. So, what we have discussed in this paper is, regardless of where you deliver the drug, either orally or nasally or intravenously, intramuscularly, or topically or in the skin, they has to go to the bloodstream when you're targeting a disease which is systemic, right? So they have to go to the bloodstream, and bloodstream is like a highway, right? Where there are lots of the drugs are going to carrying out either the blood circulation or using any of the blood component, right? And then they goes to different organ, because where the blood goes, the drug goes, right? Different organs. And what's the target? As I said, the drug, the disease is mostly associated with the cells, right? So that means the drug has to go to the cell. And some diseases are related to mitochondria, and some disease associated with nucleus. That means to get the best therapeutic effect, the drug has to go to the specific, the part of the cell, cell as cytoplasm, lysosome, mitochondria, nucleus. So for example, the disease is genetics, right? So the gene located in nucleus. So the drug mean you have to be able to deliver the drug in the nucleus. If the drug goes to mitochondria or cytoplasm, you might not get the best therapeutic effect. Again, maybe the disease is related to mitochondria. That means the drug has to go to the mitochondria to be able to get the best therapeutic effect. So just going to the cell is not the best approach to do that, right? So you see, there are lots of barrier. That mean if you think about a disease on the as a breast cancer, right? So that means the drug has to go to the breast, not the kidney, not the lung, not the spleen, not the heart. If the disease is on the kidney, that means the drug has to go to the kidney to be able to get the best therapeutic effect. So that means the drugs that is not going to the target site where they're going to the off target. That means those drugs, going resulting the cost of your treatment, and also they're going to give the lots of toxicity, which is called side effects, right? So that's just to give an example. When someone has a cancer, probably you have seen, after getting the chemo, they lose their, the body weight, they lose their hair, and then they also get other complication of blood pressure, they get complication of breathing. So why this happens? Because when someone get the chemo, maybe 1% or 2% drug are giving the therapeutic effect, whereas 90 per plus percent of the drug giving the toxicity. That means you are paying for getting the side effect, right? So that's the the real scenario of cancer therapy right now. There is nothing better. There is nothing better approach than what we are seeing. So it would be, would it not be nice to have all the drug, or 90% drug goes to the therapeutic side, whereas maybe 1% go to the off targeting side, right? So that you're gonna get the opposite effect, where you get more therapeutic effect and less toxicity. So that's what we do in our lab. So we try to optimize the formulation or the delivery system to improve or to increase the target site and decrease the off targeting site. So that paper got huge attention, and that got in the cover last year on that special issue. Uh, so let me give you another example. What happened when you, for example, you develop an insulin, right? So what insulin are supposed to be administered at? Under the skin in the stomach, right? That's the best way to get the therapeutic effect. Why we don't eat insulin like orally? Why don't we take insulin, just put on the skin? Why you have to inject, right? So just give an example. For example, so when you develop a drug, for example, a biologics, like probably DNA, RNA, or some enzyme or protein, if you inject that in your body, maybe less than 1% goes to brain. For example, a patient who has a glioblastoma or brain cancer, how are you going to deliver the drug? You cannot directly inject the drug in the brain, right? So what you have to do, you have to inject in the blood so that they go there. But there is a blood brain barrier membrane, BBB, right, between the blood and the brain. That barrier prevent the drug to go into the brain. So you get only less than 1% drug in the target site, whereas more than 99% drug are in your body, in different part of the body. So another example is, if you put a biologics, for example, insulin on the skin, on top of skin, you get less than 1% in the body, right? So that means other 99% you are paying for nothing. They're not going in the body. That means you are not getting therapeutic effect. Similarly, for example, lung tumor or liver tumor. So if you deliver a drug with a nanoparticle, only less than 1% goes to the liver, doesn't go anywhere. All the drug goes to other part of the body. For example, here, if you deliver a drug orally, for example, protein, peptide, enzyme, other large molecule drug or very hydrophobic drug orally, less than 0.1% drug goes to the body. Other drug just damage or dissociate or proteinated in the stomach because of this complex structure of the stomach, right? I'll talk about that in later details. So now talk about the CRISPR. Probably you guys know very well about CRISPR, one of the very exciting development that got the inventor got Nobel Prize this year in chemistry, right? So the CRISPR, where the CRISPR works, CRISPR is a gene editing, right, tools. So gene editing tools, that mean they have to go in the place where the gene locate in nucleus. So that means the CRISPR, to get the most therapeutic effect from CRISPR, the CRISPR has to go to the nucleus, not the mitochondria, not only to the tissue, not only to the cell membrane. So, but if you just inject the CRISPR in a human body, only less than 1% are going to the nucleus. Other 99.9% is going somewhere else. So what happened? So those CRISPR, very helpful for editing gene of the gene that you want to. That means you want to edit the gene that's the disease cell, right? Or the specific cell. You don't want to edit to your whole body's gene, right? So that means other 99% of the CRISPR that going to the off targeting, they're going to also edit the gene of unwanted cell, and that's going to give a serious effect. Probably that's going to generate new genetic disease, right? So what I'm saying is only you develop a heat molecule, and that you don't consider as a drug as long as you don't have a best delivery approach where you have less toxic effect, higher therapeutic effect, right? So so I'm going to give an example. This is very much related. This project is very much related to the current scenario of COVID-19 or coronavirus related lung disease. For example, this is a healthy lung, right? So this healthy lung, what's the purpose of lung? Lung is the body's largest filtration system. The lung filter the blood and then separated from the carbon dioxide to oxygen. When we breathe in, we take lots of air. That air contains oxygen. So when we breathe in, we are taking oxygen, and when the blood coming from all the cell carrying actually lots of carbon dioxide. So what happened when the red blood cell bringing carbon dioxide in the lung? They exchange carbon dioxide with oxygen. That means they release carbon dioxide and take the oxygen, and the red blood cell carrying the oxygen again to the cell, because the cell need oxygen to be able to produce ATP, which is the main energy of the cell, right? So that means the lung has to be able to hold enough air at a time so that our body supply enough oxygen to the amount of oxygen that need for our body, right? So if you see patient who has an asthma or breathing problem, what happened? If they walk or if they run, they feel breathing problem, because when you are walking or working or running, you need more oxygen than when you're sitting or lying down. So that means when you need more oxygen, you need to breathe more faster. But when our lung has problem, disease or inflammation, for example, these are called alveoli. You see the difference between the normal alveoli and the inflamed alveoli. So alveoli is a kind of the hole, empty hole or vacuum place where it's called the storing. They store the oxygen, right? So, for example, someone has asthma or lung inflammation or some lung disease or COVID-19, so that there's inflammation and they are not able to hold enough oxygen. So that's why we need the ventilator, right, to be able to give oxygen artificially. So this is one of the example that I'm giving you, what happened when you get the lung disease. So this is what happened, right? So when you get the lung disease, you don't breathe enough, or you have to breathe more faster, to be able to supply the oxygen that you need for your body. So, for example, those are the heat molecule, right? Those are the shortlisted molecule that consider as a therapeutic for the lung inflammation or lung fibrosis. So if you look at those those drug has been developed or used as a immunosuppressant. Why? Because if you suppress your immunity, then the progress of inflammation also slow down, right? So that the idea is, why we take insulin. The insulin is not a therapeutic. It's actually, um, excuse me. So insulin is not a therapeutic. Insulin, what insulin does is when you have enough or excess amount of blood glucose level in the body, we take insulin to be able to reduce the glucose level or sugar level in the blood. You take insulin month after month, year after year, but it never cured the disease, right? So those are kind of reducing the symptoms of disease. Similarly, those are the drug FDA approved for treating pulmonary fibrosis or any inflammation related to the lung, are mostly by suppressing or compromising the body immunity. So when you compromise your body immunity, what happened? You actually inviting the microorganism to come and host in, to be guest in your body, and that can cause other diseases as well, right? So what we're trying to do in this project is to identify a molecule that kind of be able to treat the disease, not the prevent the symptoms. So this is other real scenario. What happened? Maybe you develop 200 different drug, you push to the clinical trial. This is an example. I pull out this data from clean.gov, clinicaltrial.gov, where you can see how many clinical trials are going on for any specific disease at a time, how many drugs are in the clinical trial right now. So right now you see, there has been 200 molecule in the clinical trial as of maybe few months late, and there had been in different stages of clinical trial, as phase 2, phase 1, phase 3. So you see the green, those are phase 2, and then the gray, those are phase 1. So there are more than 200 drug in phase 1 and phase 2. But if you look at the phase 3 and phase 4, the number is decreasing. That means probably you develop something very exciting and you took the phase 1 trial, but they fail in phase 1 and phase 2. And then you have to kind of go back to the beginning to find another lead or optimize the formulation. That means as you move forward to phase 3 and phase 4, the number is slowly decreasing, and you have to kind of withdraw from the clinical trial if they don't work or they give you severe toxic effect. So when I started my postdoc in Harvard Medical School back in 2016, then my purpose of my project was to develop, or a drug that could treat the disease related to lung, the inflammation or fibrosis. So he's a MD, medical doctor, one of my co-advisor, Andrew Tiger, who passed away 2017 of pancreatic cancer. But I have been very privileged of working with him for almost 1.5 years. Anyway, so they first identified that. So there is two pathway related to the lung fibrosis. One is ROCK1 and ROCK2. So ROCK is Rho kinase pathway 1, and ROCK2 is Rho kinase pathway 2. So to be able to develop a new drug, it's very important, or that's the key of knowing the disease mechanism or disease pathology. So they started a lot of time, spent a couple of years to identify what is the pathway related, associated with fibrosis. Once they identified the pathway, so this look at this picture, the cell image. So this is the pathway that I developed. So where they found the rock inhibitor, the rock pathway is actually associated with fibrosis. And if you look at more down, where you see MRTF is a protein that convert to CTGF, which is connective tissue growth factor. So that means if in any of the state of this pathway, if you are able to inhibit, you would be able to treat the disease. Either you could use a drug that inhibit from rho GT bases to ROC, or ROC to G-actin, or G-actin to F-actin, or maybe MRTF to CTGF. So if you identify a lead or a drug that prevent or inhibit any of the state of the pathway, you'd be able to treat the disease, right? So this is called the target. Then we shortlisted or find the heat molecule of these four that is effective on inhibiting the disease in any of the states. Fasodil, which is approved by FDA in 2017. So Y27632 is another molecule, is preclinical state. CCZ1423 is another molecule in also preclinical state, and ripasodil, which is also very related to the fasodil, just in a modified version of fasodil, that also in preclinical stage. So we shortlisted these four molecules from 100 of different molecules. So this is called HTS, high throughput screening study. So where you take the cell and you create the disease in the cell, right? So for example, to be able to, so this is the fibroblast cell from the lung, and then you take the cell and add TGF beta, tissue growth factor beta. So if you add TGF beta, then cell become irritated and inflammation, and then they secrete lots of collagen. So the red, you see the red are the collagen, the blue are the nucleus, and then the purple dot are the drugs. So you take the cell, add TGF beta, stay overnight or 12 hours. Next day you will be able to see the cell. This is the normal cell in the top, and the down is the disease cell. So now you have the disease cell in the in vitro model, right? So you add the drug, those are the purple, and after adding the drug, you will be able to, if you see the drug is, the cell is going back to the normal state, that means they work, right? That means the drug has the potential to be used as a therapeutics. Good. So you select the lead, right? Select the one molecule. So you see, started with 100 molecule, finally you brought only 4 molecules, do the HTS, then identify only one molecule. So you took the one molecule. But the problem of using the ROCK inhibitor is, so if you just take the drug, inject your body, they will not only treat the disease, but also create lots of other severe toxicity. For example, encephalitis, that means inflammation, right? Anything end with itis is inflammation, right? So that's gonna create inflammation in the brain skeleton, stabilizing actin skeleton. That means your muscle and then a cytoskeleton going to be compromised, the normal function going to be compromised, and also cardiomyocyte apoptosis. That means the cell that composed of heart also going to be apoptosis state because of the rock. So that means you cannot take this drug randomly. You have to be able to modify the drug or simulate the drug to prevent of them going to random places, going them to brain, going them to bone, going them to heart, because we are targeting lung disease. So you have to develop a formulation that only carry the drug to the lung, right? So that's the key. So what is the just I what I have speaking, just I'm gonna summarize. So when you develop a drug, there are lots of limitations. The limitation one is random and specific distribution, right? So when you inject something in the blood, the drug goes where the blood goes. That means it goes everywhere. And again, the disease is very organ specific. Liver, hepatitis, the lung inflammation or asthma, the kidney cancer. So all are organ specific, right? So that means you have to be able to deliver the drug on the specific site of the organ. And many of the drugs half-life is very short. Maybe for example, TPA, tissue plasminogen activator, used for the stroke, their half-life only 3 minutes. That means you need huge amount of drug to be able to treat the stroke. um, insolubility or aqueous the many of the molecule are super hydrophobic. So the hydrophobic, that means they dissolve maybe in the organic solvent, like for example DMSO, DCM, methanol, ethanol. But unfortunately you can't inject those organic solvent in the body. So to be able to deliver a drug, the drug has to be hydrophilic or lipophilic. That means either they're going to completely dissolve in the water or maybe partially dissolve in the water. So the water or PBS are only the molecules, only the solvent that you can inject in the body. So toxicity to the healthy cell. That means for example, cancer, right? So the anti-cancer drug. So the patient who get the chemotherapy, they lose their body weight, they lose their hair, they compromise the heart function, lung function, because those drugs are not only killing cancer cell but also killing healthy cell, right? And some drugs are so sensitive to biological system. That means our body has its own defense mechanism, right? Which is called immunity. When you are taking a drug in the body actually try to prevent them to go into the systemic circulation or going, stay in the body for a long time. So our body create defense mechanism, or immunity to kind of prevent those drugs or wash out from the body. So those are the limitation of the drugs. So but how are you going to overcome the limitation? That's what we do. We kind of develop a specific formulation to be able to deliver to the specific cell, kind of decrease the dosage frequency. For example, if you need a drug to be taken 3 or 4 times a day, probably we can design a formulation that will take only you take once a day, which is called sustained release. Also, we use different polymers or different formulation approach to be able to improve their solubility, and then protecting body from the drug, because the drugs are chemicals, right? So you wouldn't want the drug damage our cell, our body's defense mechanism, our body's the healthy cell. So what we do, we protect our body from the cell. At the same time, we protect the drug from the body. As I said, body has the self defense mechanism, so that you need to be able to protect the drug, so that the body doesn't consider the drug as a the enemy. All right. So what we did in this state, angiotensin 2 protein targeting drug. So computer simulation is one of the approach where you can save a lot of time to kind of narrow down the leads. But here we did, how do you going to target the specific cell, right? The specific in the protein, because the cell membrane is most of the protein, right? And if you see, for example, the healthy cell and then inflammation cell has one protein difference than other. For example, the inflammation cell has overexpression of angiotensin 2 protein, which is the targeting of the coronavirus, right? The angiotensin 2 enzyme. So what we did, we took the angiotensin 2 protein and then we simulated to be able to structure or identify a structure of a protein that has a strong binding affinity and binding energy. So you take that, and we have been able to identify one structure of the protein. This is 7 amino acid containing a peptide that has a strong binding affinity with the enzyme, with the angiotensin 2 protein. So what we did, so now come to the formulation, right? So number one, the drug is hydrophobic, so you have to be able to make them polyphilic. For example, it's raining outside, you want to go, but you don't want to be wet. So what do? You take an umbrella or you put the raincoat on your body, right? So that you still walking under the rain, but you are not getting wet. So similarly, so for example, the drug is hydrophobic, but what happens if you put a hydrophilic, polymer on top of drug? So that means because the body's outer layer, another drug outer layer is hydrophilic, that means they're going to dissolve in water, right? And then to be able to develop a formulation with PLGA and PEZ. So PLG is lipophilic polymer, PEZ is a hydrophilic polymer. We combine together these two polymer to make a lipophilic vehicle where you have a lipophilic part and hydrophilic part. And we conjugated this protein, angiotensin 2 protein, the peptide that we identified has a strong binding affinity to the cell, and we took the drug. That the drug we found as a lead or hit molecule. So when you make a formulation with these, what happened? Because the PLJ is lipophilic, that means they doesn't get dissolved in water. So that means when you put them in the water, so the PEZ gonna be outside because they are hydrophilic, so they will be exposed on the outside, come contact to the water, and because the PLGA, they don't like water, so they're gonna be in the core of the nanoparticles, right? And because the drug is also hydrophobic, so they're gonna be core inside of the polymer. They don't want to come into the water. And the red color is the fluorescent dye. So why we are using dye, I'm gonna explain in later slide. And this is the formulation you develop, and you need controls, right? You need multiple controls to be able to compare if your formulation is better than the other formulation. So here is the other formulation where we just took the polymer or the vehicle without drug to be able to see the carrier we are developing is safe or non-toxic. And then finally, we have another control where we put the drug and the polymer, but that has no targeting, no peptide, okay? So you take the cell again, that this is the ACS, high throughput screening study, where you take different cell, the same cell line in different, well fed, where you add the plus T plus D, that means this has a drug, plus T mean targeting, D represent drug. Plus T, that means there is a targeting, minus D, that means there is no drug. Minus T, that means there is no targeting, plus D mean there is a drug. And this is only the drug, and the cell-in is another control, right? So you take all the cell, add the different formulation on the drug or the drug and incubate maybe 12 or 4 hours, whatever time. And then you put under the microscope, you see in plus T plus D, there is no protein. That means that means the drug is able to treat the disease. So this protein is a biomarker, right? So those protein doesn't, is not present in the healthy cell. They only present in the disease cell. So you see and also CCZ142, the drug also work very well. So the plus T plus D and then drug, they are similarly effective in the cellular line. So that you guys can ask a question like, why, since the drug is working very well, why we need a formulation? You will get the answer after 2 slides by yourself. So, now you remember we are developing a drug to a very specific target, which is CTZF, right? So CTZF, which is connective tissue growth factor. So that means the higher the CTZF, that means more disease state. The less the CTZF, that means that's the more normal state. So alpha SMA is another, the biomarker. This is called alpha smooth muscle cell actin. Um, Smooth muscle actin, yes. So that's, the more is alpha SMA is, that means more, that's the disease, and more CTZF also represent the disease state. So you see here CTZF and plus T plus D has the less alpha SMA and less CTZF compared to the other formulation. But the drug actually shows better therapeutic effect, the higher therapeutic effect than the formulation. That's the questioning, right? That's not what we want to see. But if you move to the animal model, so sorry, I just need to drink some water. I'll be back in a minute. All right. So here, we develop a disease model, in vivo model. So this is called preclinical studies. So you take the healthy mouse, and then you inject a bleomycin, which induce the disease or create the disease. Now you inject the nanoparticles or the formulation that you develop, or even in the free drug, right? So you see here, so the reason we added fluorescent dye is because we want to see them by our eyes. So, because we see the red color that coming from the formulation. So we added Psi 5.5, which has an excitation of 675 nanometer wavelength, so that you can see under the microscope where the drug is going. So this is one of the way of seeing the biodistribution in vitro and ex vivo, where you see when you inject the drug at day 10 and at day 14, you harvest or sacrifice the mouse and then harvest the organs. There are the key organs like the liver, lung, kidney, and spleen, where you see the plus T. Where is the plus T? That means if the particles are targeting, they are mostly likely in the lung. And the non-targeting one on the top, minus T plus D, they are not going to the lung. So they are in different places, right? So what happened, you take the tissue again, put it on the microscope, slicing, you see they are not only in the lung, but also in the cell. The key findings here is in the middle panel and the last panel, you see the difference. In the middle panel, you see lots of green. In the right panel, you don't see any green. The reason in the middle panel you see lots of green, the green represent the protein, right? The green represent alpha SMA. Because there is no drug, plus T minus D, the particle or the vehicle is going to the lung, but you don't see therapeutic effect. But in the right side, where you see the red color and no green color, that means because there is a drug, so the top particle or the formulation is going to the lung, and the drug is coming out from the formulation and giving the therapeutic effect, so that the fibrosis is disappearing as time goes, which is great, which is what we wanted to get, right? So now think about it. We have been talking about only disease cell, right? The lung composed of endothelial cell, epithelial cell, fibroblast cell, and immunocell. But when you are treating a disease related to fibrosis, you want to target only the fibroblast cell. You don't want to target endothelial. You don't want to target epithelial. You don't want to target, the immunocell. Now you have to use flow cytometry to be able to see where the drugs are going, right? So this is the way of isolating the cell. You would be able to isolate the endothelial cell, epithelial cell, immunocell, and fibroblast cell. For example, here all the pink and purple cells are fibroblast cell. But if you see the last panel where you see the light pink and dark pink, so in the left panel, which is the light pink, are the fibroblast cell that without particles, and the dark pink are the fibroblast cell with particles. So if you do the quantification, almost 20% cell with a single dose of 3 milligram per kilogram, we have been able to target 20% of the fibroblast cell. So if you want to increase the more targeting to the fibrous cell, probably you have to increase the dose. Instead of 3 milligram per kilogram, you may want to dose 5 milligram per kilogram. Or what you can do is, you instead of dosing once, you can dose once every day, right? We don't know, maybe that gonna be super toxic, maybe that gonna create other toxicity, but that's how you're gonna optimize the dose. So now, if you see the therapeutic effect, you remember? I'm going to go back to the other slide where you see the CTZF. You remember this figure, right? So CTZF here, you see the only drug has the better therapeutic effect than the formulation. But now you go to the in vivo where you see the formulation working much better than the free drug. So this is the difference between in vitro study and in vivo study. Probably you found a super nice drug that work in the cell line or in vitro state, but that might be a failure in vivo without any formulation. So you never know. So for example, this is an example of CCZ1423 drug that is super effective in vitro, but when you use in an in vivo model, they are not effective at all, but our formulation does. Our formulation been able to reduce the CTZF, almost 40 to 50%, which is a great way of developing this drug, developing this molecule as a drug. And also, because this is a super kind of deadly disease. If someone has a lung fibrosis, especially elderly people, you see the COVID-19, right? So if the elderly people get COVID-19, highly likely their rate of dying is much more higher than the healthy people or young people, right? So you see the almost without treatment, you know 90% of the animal die after 4 weeks or 3 weeks, whereas when we do the treatment, which is the red line, the line here, we've been able to save more than 70% of the animal with this formulation. With only the drug, we don't see much therapeutic improvement or development. So this is one of the way we kind of develop our formulation to be able to treat the disease. So there is another, similarly, we are also working on bone cancer, which is osteosarcoma, and also we are developing the similar formulation for bone regeneration. For example, someone do an accident, fracture the bone, probably that takes maybe 1 to 2 months to heal naturally without any treatment. But we have been trying to develop formulation to expedite the healing process, maybe within a week or two. Especially, this is other project that we are doing in our lab is uh, developing drug for arthritis, or the which is called joint pain. So you see you have been able to, when you inject something through tail vein, it doesn't go to the lung, it doesn't go to the heart or anywhere, it just go to the the inflammation site on the leg. uh, so this is, I'm going to give an example of CRISPR. You just use a CRISPR, it goes to everywhere in the body, probably sticking on the surface of the membrane, cell membrane, it doesn't go in the nucleus. So we develop cationic polymer. So this is a polymer developed with MGP, methoxypolyethylene and serinol, and then finally we did a MPG with the PLL, polyl lysine. That if you add the polyl lysine with the CRISPR, because this is a cationic polymer and CRISPR is anionic molecule, they form a nano complex, nanoparticle, and that has a super binding affinity to the nucleus. So this is other example here I'm going to give. The nabitaclox is another drug that got FDA approval maybe 2019 for cancer therapeutics, but that drug work in the mitochondria. So that means if you want to get the best therapeutic effect, you have to be able to deliver the drug to the mitochondria. So we developed another formulation, which is linked with TPP, triphenylphosphine. So the triphenylphosphine linked with the polymer has the super binding affinity to the mitochondria. So that if you use this vehicle, you'd be able to deliver the drug to the mitochondria instead of nucleus or cytoplasm. So just to, if you look at the cell, for example, the doxorubicin goes to the nucleus, if you just deliver without any formulation. But if you use doxorubicin with our formulation, they convert their location to the mitochondria instead of nucleus. There is other example, you see here, only doxorubicin, mostly 99% in the nucleus, but doxorubicin is the formulation, the nucleus in the center of the cell, but you see in the last panel where you will see they are no more in the nucleus. So this is an approach we also are considering to use to overcome the drug resistance, because when you get the first chemo, it gives super good therapeutic effect. As you go second, third, fourth, the therapeutic effect get reduced slowly, because the body, the cell get resistant with the drug. So the most of the resistance happen in the nucleus. So we are thinking if we deliver the drug to the mitochondria instead of nucleus, probably we would be able to improve the therapeutic effect without changing the drug, right? So just we are, this is the in vivo model where we took the tumor model and been able to reduce the tumor size within 2 months. So again, it's not only about the how fast you can treat the disease, but also important is can you treat the disease without damaging the body? That's more important for us now, for especially the cancer treatment. It might take 2 months, but without damaging any major organs, for example, heart, lung, kidney. So I'm going to give an example what happening now with the nanoparticles. Up to 2016, there have been 25 drugs got approved with the polymeric formulation approach. There have been 45, other nanoparticles were considering in different stages of the particles. In the past 3 years, 75 new clinical trials started in FDA. Before FDA approval, and only last year there has been 15 new formulation, nanoparticulate formulation enter into the clinical trial. So with this polymeric nanoparticle approach, we are making significant progress overall the nano medicine community. So in my contribution, like we've been able to make a oral GLP-1 formulation that formed a KB Biomed startup company in South Korea, where I was a co-founder and chief scientific officer. So now this is in phase 2 clinical trial. Science is during my PhD work at University of Utah, We develop ileum targeting drug delivery approach for protein and peptide delivery. That's also a startup in based in Salt Lake City in Utah. And I2O, which is injection to oral delivery therapeutic approach based in Boston, Harvard spin up, where I contributed significantly on the diet induced obesity and oral insulin delivery project. So those are in different state, and this is I'd like to thanks to my great mentors. Without mentor we are nothing. We need mentorship all the time. It doesn't matter what state you are in your career, either student, professor, or even senior professor, it doesn't matter. So I am always thankful to my mentors. What I am is a result of my mentors. So Samir Mitragoti and Jason McCarthy are two great scientists and professor at Harvard. I worked with them almost 4 years before coming to Texas. Yongki Lee, I have been with him for more than any other people, uh, you know, um, except my wife and my parents for sure. So I've been with him for 6 years during my master's and PhD. Yuhan Bey, a professor at Utah, I've been with him for 1 year during my PhD. And Kang Moho, I have been with him for almost 1.5 years. So, I have been working with the people from different background, for example, chemistry, chemist, chemical engineer, material science, biology, and different. So that helped me to understand about the different state of drug development and drug delivery system. And right now I have a lab where I, manage them and kind of work with those young, outstanding folks. 3 PhD student. Isaac actually just graduated a few months back. Huda and Afrin are outstanding, rising star in my lab. They are doctor of engineering student, actually, not pharmacy, but those four, Marlene, Manuel, Clarissa, Jasmine, are doctor of pharmacy students. I have two research technician and a couple of undergraduate students. And this is only my 14 months, but still I have, kind of 14 people working in lab, but it's growing. I'm hoping to have a lab of 20 people where maybe 7 or 8 PhD students. And my lab is funded by the university, by the graduate school and foundation as of now. Uh, by saying that, probably I'm running out of time, but let's spend as many time as you need for answering your question if you have any. Thank you again to the organizer and also the people for attending to my talk.